Hello and welcome to KognitoKey. Today we're diving into something truly fascinating from the world of plant science. Just imagine taking a tiny part of a flower and growing a whole new plant from it. Sounds like something out of a sci-fi movie, right? Well, guess what? It's totally real, and we're about to find out exactly how it works.
So let me just ask you, what if you could completely skip the seeds, the soil, all that waiting around, and just poof, clone a plant straight from its flower? I mean, think about it. This isn't just some wild idea. It's the real deal behind a super powerful technique we're going to break down right now.
All right, first things first, let's get into the what. This amazing process has a fancy scientific name, gynogenesis. Now, I know it sounds a little intimidating, but don't you worry. We're going to unpack exactly what it means and why it's such a huge deal for the future of plant tech.
So here's the core idea. Gynogenesis, or what scientists also call ovary culture, is basically a lab technique. In vitro just means in glass. So picture everything happening in a super clean, controlled lab setting. What they do is they take an ovary from a flower that hasn't been pollinated and pop it onto this special nutrient-rich gel. And the result? A brand new haploid plant. All that means is it has just one set of chromosomes, making it a perfect little clone of its mother plant.
And you know what's cool? This isn't some brand new, just discovered science. Nope. This whole field was kicked off way back in 1976 by a scientist named San Noam. His work was the big breakthrough, the foundation for literally everything we're talking about today. It's just amazing how one person's discovery can set off a chain reaction of innovation for decades, isn't it?
Okay, so we've got the what down. Now for the really fun part, the how. How in the world do scientists pull off such a delicate procedure? Let's take a peek inside the lab and walk through it step by step.
Honestly, you can think of it like a super precise, very clean kitchen recipe. First up, they gather healthy flowers and give them a good wash. Step 2 is a big one. It's absolutely critical. They sterilize the flowers to get rid of any nasty germs or microbes. Then, inside a totally sterile little box, the ovary is carefully separated. Step 4, it's placed onto that special nutrient gel. And finally, step 5, it's put in a cozy, warm spot at 25 degrees Celsius, and that's where the magic really begins.
Now this is where it gets really interesting. Even once it's in the lab, the ovary can go down one of two paths. To kick things off, that's the induction phase, it's put in a liquid nutrient bath in total darkness. But then for the next step, regeneration, where it actually grows into a little baby plant, it's moved to a solid gel and gets a good 16 hours of light. See that? Just a few simple tweaks to the light and the medium, and you get completely different stages of growth. How cool is that?
Okay, so we've covered the what and the how. But maybe the biggest question of all is why? I mean, why go through all this trouble? Well, let me tell you, the reasons are incredible. The real-world applications of this are a massive game changer for both science and farming.
I mean, just look at what this technique lets us do. Scientists can literally watch an embryo develop from the very beginning. They can see how different plant hormones work. They can create new hybrid plants way faster than before. We're talking about new types of crops that can survive a drought or produce more food. They can even study pollination itself up close. It's just wow. Each one of these is a huge deal, and it all comes from this one technique.
Let's zoom in on just one of those amazing applications, something called polyembryony. It basically means many embryos. So instead of getting just one plant from one ovary, scientists can get it to sprout multiple shoots. We're talking 10 or even more. It's like a copy machine for plants, letting them multiply the best ones really fast.
Now, as exciting as all this is, we've got to be real for a second. It's time for a little reality check. This technology isn't perfect, not by a long shot. There's some pretty big hurdles that scientists are still trying to figure out. So let's take a look at the challenges.
So the number one biggest problem? The success rate. As you can see here, it's really low. We're talking only about 1 to 5% of the ovaries actually grow into a plant. That means for every 100 attempts, you might get nothing from 95 of them. And that right there is the main reason this isn't used everywhere just yet.
And that's not all. This whole process takes some serious technical skill. Yeah, it's definitely not a DIY project for your kitchen. Plus, it doesn't work on every single plant out there. So far, it's really only been proven to work well in big-time crops like wheat, rice, maize, and sugar beet. Getting this to work for more species, that's the next big frontier for researchers. So all of this leaves us with a really exciting question, doesn't it? Even with all the challenges. Just imagine if scientists could perfect this technique. Could this be the key to designing the super plants of the future? You know, crops that can handle climate change, that are packed with more nutrients, and that can help feed our growing planet? It's a powerful thought.
And on that hopeful note, that's all we have for you today. I really hope you found this as amazing as I did. If you enjoyed this little journey into plant science, do me a favor and hit that like button, share it with someone who loves to learn new things, and of course, subscribe to KognitoKey. We've got so much more to explore together. Thanks for watching.